morning, everyone. Assalamualaikum. My name is Tengku from the Department of Ophthalmology and UMI Research Center. It gives me great honor and privilege to be introducing our visiting Fulbright scientist who's just landed yesterday, who will be with us for a month, Dr. Samarjit Das, who's also my project collaborator. We've known each other for over a decade, and we've worked on productive projects together, and he's here today to talk to us about introduction to intracellular social media. So there's a lot of cellular mechanistics introduction to us, especially as clinicians. So I'll pass over the floor to Sam.
Oh, thank you so much, and thank you for waking up this early, and thank you, Professor Kamalden, for nice introduction. As she mentioned, I just arrived, and I was fortunate enough to have good food last stop for this talk. And today's talk, I'm going to discussing about social media, and that's the topic. It's very easy for everybody because everybody use some forms of social media every day. And today I'm going to connect you with in our human body's communication between one organ to another organ, or one cell to another cell, in forms of a tweet or a Facebook post or Instagram, whatever is your favorite social media. I want you to just think about it the entire time. We will talk about all those difficult names to memorize. So this is my disclosure slide, and I will be talking to some extent the SOGNOF initiative, which we just developed during the COVID. And the objective of today's talk, obviously, to give you some introduction about extracellular vesicle and how they may play important and critical role in several human diseases. And also we will discuss mainly about the diagnostic platform where we can use these information very easily to develop a diagnostic test. So it is very open to anybody and everybody who is interested to know about disease and who also are thinking about implementing these information to the therapy. So let's get into this. This is a very common thing, whoever use Facebook. So this gentleman over here, he posts a picture, and it says something about his status, something about his COVID status, and all of these people over here can read the post, what he did, and they immediately know that this person got vaccinated. Even though he's vaccinated, he still thinks the wearing mask is a very good thing, and also what kind of vaccine he got. So this is, if we consider this picture or post as an extracellular vesicle, and all the information I just did to you, like the vaccine or wearing mask is a good thing. So these are the cargo. And so now the entire talk, what I'm going to tell you about, just think about this, and we will talk about RNA, DNA, protein, doesn't matter. The bottom line is, this is a post, and we will dig the information about this extracellular vesicle. And this extracellular vesicle comes from this person. So I know about that particular cell, and wherever it will go, whether that can provide. In that last part is, if this gentleman goes to a party, we already know that he's vaccinated, chances of spreading out COVID is very less. So that's the extracellular vesicle cargo can do to other cell types. So organ-organ communication, and that is the premise of intracellular communication. So let's talk about server. I talked about server. There are 3 flavors of server each cell secretes out. So the biggest one is called apoptotic bodies. When an organ doesn't want to belong, they will say, we are done, we are just going to destroy our cells, they secrete these bigger than 1 micron sizes bodies, and these contain everything, whatever a cell has, like nucleus and RNA, DNA, mitochondria, things like that. So you can detect DNA, RNA, protein, everything in this apoptotic body. One good example is lactating mother. When they finish the periods of lactation, so what they do is they just destroy those cells. And apoptotic body is one of the form. And then the minimum size is microvesicle. Microvesicle, they do not use any cellular energy. They just bleb out from the cell. Therefore, it can contain RNA, DNA, as well as protein, whatever the cell does not need. And micro particle formation is kind of severe stress when the cell does not have enough energy to form something, ATP, for example. And also the smart cell, for example, cancer cell, they don't like to use energy. So they just bleb out from the cell membrane, and they ultimately, they secrete all the materials of the cell. But today we are going to talk about the smallest extracellular vesicle, which is about 30 to 200 nanometer in size, and they are called exosome or small extracellular vesicle. And in our experience, these extracellular vesicles, they do not carry any kind of DNA. So they carry RNA and protein and lipids. And so the reason behind it is they use intracellular energy, forms of ATP, to invaginate cellular membrane and stay kind of in the cytoplasm until it secretes outside the cellular vesicle. Therefore, they do not have any contact with the nucleus. And that's why we believe that we do not have any DNA in them. But the size is one of the criteria of differentiate between three different extracellular vesicle. However, exosome is the most popular extracellular vesicle in terms of human disease, diagnostic, and other purposes. The reason is we have so many tools available. We can use the markers protein called tetraspanin, as you can see here, CD9, CD81, and CD63. And also I talked about ATP-dependent biogenesis process, because ATP energy control the entire production. So therefore, cellular function or how cell are doing depends on the exosomal biogenesis. They carry protein, talked about it. They have some receptor outside, and we will talk more about that, and that's very important. And cell-specific marker, which is the premise of today's talk, and we will talk about why a cell like to secrete extracellular vesicles, especially exosome, and the reason behind it, it's the protein control. When cell under stress or any kind of stimuli exposure, cell try to maintain regular cellular homeostasis. So therefore, they want to secrete something out. They want to secrete those extra material which causing more stress in the cell. Those are the main thing. It's very similar your daily life. You generate so much trash, and if you want to keep it inside the house, it will cause problem. It can smell or things like that. So therefore, you take those trash out. Cell does the same thing. And here are some of the fancy words, but these with nothing but the trash for the cell. This is a cartoon representation of today's talk. And I know it's a busy busy slide. What you can see here, my pointer, and that's the donor cell. That means this cell has some extra RNA, DNA, and protein aggregate inside the cell, want to decrease the level. Therefore, they use a stress or stimuli, which is an ATP-dependent manner, and that can cause an invagination on this side, and that form an early endosome formation. And that early endosome formation use ATP called Rab5, and they form a multivesicular body. This is like our trash can inside the home. And every day's trash goes inside in a smaller package, smaller bags, and those are the multivesicles inside the multivesicular body. And then they again use a ATP called Rab27. They fuse this multivesicular body back to the cell membrane and then expose those smaller particles. And that's what goes out through through these pores formation into the extracellular space, and these are circulating. And from this cell, now it traveled to this cell by association of ligand receptor. It delivered the cargo, and this cargo is active now. So what I want you to point out here is this blue thing, this cartoon shape. What we did is X. So what we are trying to say is in this cell, X is a unique protein or ligand present in the cell membrane, which incorporate into these extracellular vesicle, and we will got our cell-specific extracellular vesicle. So this is our responsibility to identify this X in the circulation. If we can find the X, then we will find about what is the trash in these cells. So you can use at this form as basically a diagnostic, early diagnostic biomarkers. And if you find it here, this X goes to this cell, and we can find the pathogenesis of a disease. And that's what we will talk about throughout the talk. And again, you are familiar with this person, and we met about more than a decade ago. And Dr. Kamalden, at that time, we started our version 0 biomarker. At that time, we did not know much about extracellular vesicle. It's a new thing. Exosome, forget about it. We did not know what's the best way. And it was challenging for Professor Kamalden here because she was constantly communicating. At that time, remember, there was no Zoom, nothing. So we used to do a Skype call and figure it out what she found in the lab and how she, what would be the next step. And then I do those work in my lab using animal sample and tell you, try this. And this was a circle, and the circle is still going on, fortunately. So this is a University of Malaya sample. We collected here, and then she processed it, and we performed a microarray. At that time, RNA sequencing was not that popular, or also probably in our pocket, we did not have enough money to do that. So we did a global microarray approach, and we identify these red dot representation of microRNA, which are upregulated in type 2 diabetic Malaysian population. And healthy control is these green dots, which are upregulated in healthy control and downregulated in type 2 diabetes. So this kind of a global profiling we perform from this sample, which were collected in University of Malaya. And so what next? So during that time, it was challenging to develop a biomarker assay without a normalization strategy. So in collaboration with Professor Kamalden, we developed a panel where we have a bunch of microRNA, and we were throwing one microRNA at a time to find the best strategy to normalize our data. And I don't want to go into details why normalization is very important. Everybody in this audience knows more than I do. So we want to make it a more clinically relevant approach. And as I said, if we can collect these extracellular vesicle over the time and going back longitudinal study at the earlier phase, we can detect it very early. So I thought Professor Kamalden's model was excellent to kind of go into the diabetic progression, because none other diabetic vasculopathy, we have this privilege to collect image at different stage, what we can do with the eye. So here we have 4 groups: control, type 2 diabetes, no complication, type 2 diabetes, non-proliferative diabetic retinopathy in the reversible phase, and then proliferative diabetic retinopathy. And so we collected all those information, and based on this information, we thought, what would be the approach to fix the normalization issue? And we thought, oh, red blood cell, sorry, I don't know why it went up, but what I was pointing out, the red blood cell count number. And as you can see, the number of red blood cell is not changing across the board. And we thought then whatever microRNAs are red blood cell marker, we can use those as a normalization control because they are not going to change across the four groups. And that's exactly what we did. And we found a microRNA 15A. As you can see, this is an interesting slide. I will go into details, but just for 2 seconds, believe me, that it goes up throughout the non-proliferative diabetic retinopathy. And you see this is a progression of microRNA in the 15A in the circulation. So therefore, we can say it's not only a diagnostic biomarker, but it's a prognostic biomarker. Now, those people who were believing me for 2 seconds and they were talking about proliferative diabetic, what happened to the 15A expression in the circulation? It's come closer to the healthy control. And that was the time I appreciate the concept of cell-specific exosome. The reason we at that time, obviously with the help of Professor Kamalden, we figured it out is most of these people or all of these people are taking exogenous insulin at this stage, or even some of the people here. And because of the insulin taking, we believe that pancreatic beta cell is the key factor in this whole 15A story, that 15A is secreted by pancreatic beta cell. And on the basis of that, we developed a mouse model. But in this slide, what I'm going to show you, that when a person gets hyperglycemic with this extra glucose in pancreas, they send signal to produce more insulin. And when they produce more insulin, microRNA 15A plays an important role in that particular transition of adaptation, not transition, adaptation of producing more insulin. And 15A is a byproduct of that process. And therefore, there are lots of 15A getting stored in pancreatic beta cell, and they secrete out in forms of these exosomes, and they start delivering, or they start going all around, including retina, and cause various vasculopathy, such as diabetic retinopathy, cardiomyopathy. And here we mainly focused about the retinopathy, but it can also lead to cardiovascular myopathy, cardiomyopathy. So this is initially our goal was to kind of focus on diabetic biomarker, but now you open the Pandora box and we figured it out. Oh, it's not confined into one disease, but if we play this platform more wisely, we can go into details in different disease condition, and we can assess the early diagnostic part. And so since then, we started working on a huge manner to identify several diseases, including diabetes. And here we talked about two wonderful ladies, Fernanda and Shushmita. In fact, Shushmita and Dr. Kamalden, they kind of overlapped when Dr. Kamalden went to my lab as a Fulbright scholar, and we had a nice time working there. But let's focus on this particular slide, and I will tell you so from that diabetes story, how we went to in a global picture of obesity. Obesity, here we are talking about two particular groups, BMI, and these samples are collected in King's College London over the time. And these samples are one we call healthy, which is BMI less than 25, but obese, BMI 30, more than 30. And these individual is, at that time when we collect the sample, they were not diagnosed with any kind of complications such as diabetes, hypertension, or fatty liver disease, or there was no complication. They are just obese. So we collected their blood and we looked at their exosome profile in terms of the number. There is no difference. And then we also looked at their RNA profiling, like whether they have any RNA, like one RNA speaks out, hey, look at me, or something like that. But we did not. We perform even molecular thing, and then we can show you here that two microRNA, green segment and red segment, red are upregulated in 30 BMI and down. But those microRNAs is very difficult to follow, like our microRNA 15A, where there are several organs they express microRNA 15A. So how can we go into these red dots or green dots? And so at that time, the concept of liquid biopsy of pancreatic beta cell. Here, I'm just going to transition from whatever Dr. Kamalden and I, we started working and keep working on it, which is pancreatic beta cell specific, capturing a strategy to capture pancreatic beta cell specific exosome or small extracellular vesicle and detect what is in there. So exclusive rights, I would say, with collaboration aspect for microRNA 15A is with Professor Kamalden here, but we are also working what are the others marker, because now we are outside the umbrella of type 2 diabetes. We are even one step behind, which is obesity. And so we came up with as you know, pancreatic beta cell primary culture is very difficult. No donor we will find. So we collaborate or at an MTA with the Insurem, because they develop a human pancreatic cell line from a 62 years old female without any complication, and we culture these in our lab. And just important to note, FBS, when you develop a platform like that, FBS is your not a friend, because FBS will contaminate your exosome or your patient or your subject's exosomes with cow exosome. Therefore, it's very important to come up with a strategy where you are not going to use FBS, or you are going to use exosome depleted FBS. Anyway, so these are the steps in a small slide. By basic science people will appreciate each bullet point takes months, but for clinician, this is how we develop a cell specific exosome capture strategy. And the thing first, we start with proteomics to see what are the protein expressed in these exosomes. Then using bioinformatics, we identify out of those list of protein, what protein present only on the surface of this exosome. And then we validate using Western blot. And this is one of the example of the last step, Western blot of those three markers: PC13 GIP receptor, GLP1R receptor, because these are the positive hits for proteomics from that human pancreatic beta cell culture. After proteomics, and we validated it's only GLP1R. I'm giving you one example, what Shushmita did at that time. And now we kind of try to look whether in the mouse model, because in human sample, it's very difficult through all IRB approval, and it takes months. So mouse model is very easy, in terms of the sample. And so we kind of validate all the exosomal markers, spatially the GLP1 are also present in mouse exosome. And mean6 is a cell line of pancreatic beta cell from mouse. And then we develop a method. I will show you a cartoon how we capture cell-specific exosome, and we call this method as ICE, immunocapture of cell-specific exosome. And here are certain markers, well-known markers. MicroRNA 375 is a marker for pancreatic beta cell, and you see the capturing with GLP1 positive exosome gives us a very high abundance of 375. And 451 is a red blood cell marker. 223 is a platelet marker, and I cannot see the last one. 150 is a white blood cell marker. So therefore, yeah. Can I go? Yeah.
Lower this.
I did.
Oh yeah.
Okay. So 150 is a white blood cell marker, and therefore, what happened here is this method, whatever we generate, shows a unique opportunity to interrogate cell-specific exosome. And so, yes. And so now we looked at 15A, what Professor Kamalden found in her initial study. Now we want to take that information and see whether in a mouse model, if we feed the mouse a high-fat diet for 18 weeks, can we capture their by beta cell-specific exosome, and can we see the increase of 15A in there? And indeed, this color bar shows that. However, we see a slight increase in the non-pancreatic beta cell specific exosome. And that is the challenge we still have, and we have a way to kind of eliminate that, and which is 15A also expressed in other cell types, such as leukocytes. And with a diabetes or hyperglycemic condition, those cells also can change or regulate 15A, and those will be contaminated in your pancreatic beta cell specific exosome population. So yeah, we are still trying, but I will show you positive result. And so then Fernanda and Shushmita, they developed ICE, which is a multiplex approach. And so what I'm talking here, so this is blood you collect in your clinic. Then we isolate plasma. From plasma, we have a way to isolate small extracellular vesicle. In this case, we use ultracentrifugation or differential centrifugation method. And then we generate these plates. These are special plates, and we have a way of capturing cell-specific marker. So for obesity project, what we did is we capture beta cell specific here in the plate 1. Whatever is unbound, that means did not stick to the plate, we take that fraction and use it in a endothelial cell-specific plate, capture them, and then the same material which was not stick to the plate, we took it and put it in the adipocyte specific. And then the fourth one right now is hepatocyte specific. So individual cell type, we went through this year-long method of identifying what would be the marker, how to generate the ICE capture, and then we go into details. Let's take to the beta cell. Now we are coming back from mouse to human, and those are the sample which we collected in King's College London. And these are the exosomal parameters. This method of characterizing exosome is called nanosite or nano tracking analysis. And you can see the size of those exosomes and the concentration in those blood. And then obviously we checked our GLP, whether they are present in the exosome, lysed exosome from the sample, as well as on the surface by flow cytometry. And then here is the distribution. Again, it took us 4.5 or 5 years to identify each of these, counting the number of exosomes present in these samples. For example, beta cell specific exosome, endothelial cell specific exosome, adipocyte specific exosome, hepatocyte specific exosome in the same sample. So sample volume is important for everybody, including clinician as well as basic science translational people. Giving 1 ml of blood is a huge deal in certain cases, but here we are talking about 400 microliter of plasma, and we will interrogate 4 different cell type specific, and that is the platform we developed. And so now we will go. Remember, I just want you to remember, from your memory for like 5 slides back, there was no difference in the exosome number between BMI 25 and BMI 30. But now if we isolate pancreatic beta cell specific exosome, we see a statistical significant difference, almost double the number pancreatic beta cell when the BMI is high. So whatever the cargo inside can be showing that the stress of pancreatic beta cell. Similarly, endothelial cell specific. Interestingly, there is no difference in the cargo number, exosomal number, but I will show you some characteristic. Hepatocyte, yes, we see an increase in number, and then that kind of relate to the fatty liver disease. And adipocyte specific, obviously, this is a no-brainer because obesity is, you have a lot of adipocyte, white adipocyte, and that's why the number will be more cell, more exosomes, and that's the rule of thumb. And now we will go into each individual cell type. And this is a pancreatic beta cell panel. As you can see, we call it pan-exomer. Pan-exomer will give us combination of these 8 microRNA. Again, remember here we do not have 15A, but 15A, as I said, It's professor's right. So anyway, 8 of these with combination, we can talk about pancreatic beta cell health. And based on the label, right now, this is a qPCR method, but now we have a digital PCR method. And so we can absolutely quantify each microRNA's expression, and that will tell us what. So for example, we can tell you that in coming 3 years, this person will develop type 2 diabetes, or coming 5. We need a lot of data, machine learning approach to answer that, but we are on very good position to go there. And we can also talk about, even though this person does not show any signs of type 2 diabetes, but this person has a huge need for exercise or even metformin, and the clinician can play by the day, by the month or week. They can say, hey, look, I got this result. Why don't you come back sooner than 6 months? And we will do it. And maybe at that time, you can diagnose as a pre-diabetic cases. So yeah, I don't want to go individual cell type, but this is the panels what we developed. And obviously pancreatic beta cell, we send you the list. These are, we call it panels. And based on this, the risk factor, we can determine how soon this person will develop hypertension or how soon the fatty liver disease. And so that's the story of obesity. And this is the what I talked about. And then last few minutes, I know I'm running out of time, but I will talk about the one person can use this and go into a clinic very easily, and that's exactly what we are doing. We are running out of time. Okay, so this is a COVID project. I don't want to go into details, but I talked about all of these things. But this is a quick demo I want to show where Fernanda's hand can be shown, and then this is the device. Right now we have a more better device, and you don't need, for example, ultracentrifuge or anything like that. We made a microfluidics chip where you spit. In here, they're using pipetter, but you can just spit directly onto there, and then that's the reader, and the color of the light you see on here will tell you the basically whether the processing, sample processing, and this device is generating heat and electricity to separate the exosome. So therefore, all the pain of isolating exosome using ultracentrifugation will be done in 2 minutes, and that's what she's waiting for. And I don't want to go into all video here, wait time. We go pass, and then when you open, you see 2 colors. If red light comes, if the green light comes out, then you're COVID negative. I know it will come red, but still I want to show you. And you see the red color here. So anyway, and this is the version what we have, and this is what you have to do at home. It's under FDA consideration. You turn the machine, you spit, put the machine in, reader in, and then you see positive, negative. And now this machine can speak for the blind people that they can do at-home testing very easily. And thank you.
Thank you so much, Sam, and congratulations on those kids, especially in getting the FDA approval. In the interest of time, we're running a bit late. I think I'll leave the floor to the next chairperson and speaker, and we'll take questions if we have time then.